to Everyday Science. Today we're going to sample tissue from plants. So the reason we're doing this is we actually have to genotype them. And genotyping means is that we are going to be isolating genetic material from the plant tissues. So in this case, I'm going to be harvesting flowers or leaves, and we are going to isolate that genetic material, and we are going to check it for different mutations. So I have a variety of plants here. Each of them have different mutations, and I'm just checking to make sure that those mutations are what they're supposed to be. So to do this, we are going to take one of our plants. So I've got a plant up here, and what I'm going to do is I'm going to take some of the tissue. So let's see if I can do this. Sorry for the shaky cam, everybody. And I'm going to just take my tube like so and put some flowers in it. Okay, like so. And I'm just going to use the cap to just pinch it off. Ta-da! Okay, so here we have our tissue. All right, now I have to make sure that this tissue does not degrade. So what that means is that I need to make sure to freeze it so that the DNA molecules, which is the genetic material, stay stable so I can work with them later. So this isn't as simple as just putting it in the freezer. We actually have to do something more extreme. So instead, we are going to be using liquid nitrogen. So I have my container here. There's liquid nitrogen in there. So liquid nitrogen is just nitrogen gas, but it's been compressed and it's very, very cold, like super, super cold. So we're going to drop this guy in here, and you can hear the sizzling. And so that flash freezes the tissue. Now, as the liquid nitrogen warms up, it starts to evaporate. So you can see a little bit of smoke. And so what I'm going to do is just go like this. Ooh, right. So it is super cold. So that was just a little bit from my breath causing the liquid nitrogen to evaporate. So I can actually not touch it with my hands at all. I'm going to have to use a special kind of pliers to pull the samples out later. But this allows me to freeze a lot of tissue, put it in a very cold freezer, and sort of a deep freeze, and then analyze it later. So that is what we're going to do. And so maybe a later video I will show you how we isolate the genetic material from the plants. So I hope this was interesting, and I will talk to you guys later. You can find me on Twitter and Facebook and my website, which is drunkphytologist.wordpress.com. And so I will see you around. Like, comment, and subscribe, and I will talk to you later. Bye!